The physical environment of a living cell is chaotic, volatile, and difficult to control. Digital code, by contrast, is absolute. For over a century, immunology relied on the physical rules of biology, building upon the foundations established by early pioneers like Louis Pasteur. To design a vaccine conventionally, the pathogenic microorganism must be isolated and cultured in vitro, grown in a physical laboratory. Only then can its components be extracted. But if it cannot be sustained in a petri dish, the pipeline halts entirely. Even when cultivation is successful, rapidly mutating pathogens like HIV or hepatitis C introduce a second failure condition, hypervariability. By the time a physical culture is isolated and developed, the virus in the host has often mutated its outer structure. A third failure condition occurs when pathogens possess surface proteins with high structural homology to human proteins.
Injecting these cultivated antigens risks triggering autoimmune reactions, where the body attacks its own healthy tissue. The conventional pipeline repeatedly hits a dead end because it is entirely dependent on the physical limitations of the live pathogen. The solution to this cultivation bottleneck arrived with the genomic era, in the form of a computational approach known as reverse vaccinology. Instead of attempting to grow a physical sample in a laboratory, this method begins entirely in silico, operating directly from the pathogen's fully sequenced genome. A physical culture only provides access to antigens expressed at extraction. In contrast, the raw genomic sequence contains the blueprint for every possible protein. Algorithms scan this data to deduce vaccine candidates before a live microbe is ever handled. This digital process does not bypass the need for animal testing later in the sequence. What it replaces is the unpredictable physical discovery and isolation phase. Moving the initial discovery phase into the digital realm allows researchers to evaluate potential antigens based on their genetic blueprint, bypassing the physical constraints of the laboratory. The primary objective of this in silico pipeline is to sift through millions of raw base pairs to locate open reading frames, or ORFs, that encode surface-exposed proteins. As this animation demonstrates, the raw DNA sequence is mathematically bracketed into these discrete ORFs, which are then passed into a sequence of algorithmic filters. The first gate uses a tool called pSort-B to predict cellular localization. Researchers discard any proteins destined for the interior of the pathogen, as the immune system will not encounter them. Next, the remaining sequences enter the Signal-P algorithm. This scans the data for specific signal peptide cleavage positions, confirming the protein is actively transported outward to the cell boundary. They then pass through TMHMM, an algorithm that calculates the sequence's hydrophobicity, its repulsion to water. A high hydrophobic spike indicates the presence of a transmembrane helix, meaning the protein is anchored in the cell wall. Finally, if evaluating gram-positive bacteria, a lipo-P gate specifically isolates lipoproteins, distinguishing them from other membrane helices. Through successive bioinformatic gating, these algorithms reduce an unreadable ocean of raw DNA into a finite list of structurally confirmed surface targets. Identifying a surface protein is only the first step. The algorithm must now locate the exact epitope, the specific structural pocket on that protein that our immune cells physically bind to. Predicting a B-cell epitope involves distinguishing between a linear epitope, which is a simple peptide sequence, and a conformational epitope. Conformational epitopes require advanced modeling tools, like Discotope, because the binding site itself does not exist in a flat sequence. It only emerges when those amino acids fold into a specific three-dimensional architecture. To clear hypervariable viral infections, researchers must also map T-cell epitopes, which govern the body's cell-mediated immune response. This introduces a biological constraint. A T-cell can only recognize a pathogenic peptide if it is physically presented by the host's own MHC class I or II molecules. This diagram illustrates how the Epimatrix toolset operates to predict binding affinity across over 100 different human MHC alleles. A sliding window brackets exactly 9 amino acids at a time, moving step by step down the target sequence. The algorithm calculates a score for each resulting frame on a standardized scale. A massive spike identifies the exact sequence most likely to serve as an immunodominant trigger. These predictive algorithms confirm the chosen peptide sequence is physically accessible on the pathogen's exterior and provide a mathematical score for its ability to trigger the human immune system. A distinct vulnerability remains. If researchers sequence the genome of a single, isolated pathogen, the resulting vaccine might only be effective against that exact strain. To counter this, researchers employ a modification called pangenomic reverse vaccinology. This schematic visualizes the logic. The algorithm cross-references the genomic data of multiple different isolates of the same organism simultaneously. By scanning across populations, the system isolates orthologous gene clusters. These are surface epitopes so evolutionarily conserved that the pathogen cannot mutate them without losing viability. Extracting these universally conserved sequences yields a pangenomic target, ensuring the resulting vaccine will neutralize an entire species population. The definitive proving ground for reverse vaccinology occurred against serogroup B Neisseria meningitidis, or MEN-B, a lethal cause of bacterial meningitis. For decades, classical vaccinology failed to produce a viable defense against this specific pathogen. This structural comparison highlights the roadblock. The outer membrane proteins of MEN-B share a near-identical sequence to human neural tissue. Introducing cultivated MEN-B proteins as a traditional vaccine carried the risk of an autoimmune reaction. To bypass this, the entire genome of the bacteria was digitized and fed into the reverse vaccinology filtering pipeline. The physical tools of biology had reached a dead end. If a non-reactive surface target existed, it had to be found buried inside the digital code. The algorithms identified novel, surface-associated lipoproteins within the MEN-B genome, structures that had never been observed in decades of traditional laboratory culturing. This diagram outlines the transfer back to the physical world. Through recombinant protein expression, the digital DNA sequence of the novel lipoprotein was inserted into an E. coli plasmid. The E. coli was used as a biological manufacturing unit, physically producing the exact outer membrane proteins predicted by the algorithm. Scientists then used physical assays like ELISA and FACS to confirm that the E. coli was successfully expressing the predicted MEN-B targets. Those computer-generated proteins were isolated, formulated into a vaccine candidate, and entered Phase 3 clinical trials, successfully inducing protective immunity in human volunteers. By trading genomic data as a searchable inventory, reverse vaccinology allows us to engineer immunity even when a pathogen cannot be grown in a lab.